Good morning, everyone. Buenos dias. Unfortunately, I don't speak Spanish, so you may want to put your translator headphones on. Thank you, Sophia, for having me here. As Sophia said, my name is Tony Favello. I'm the marketing manager for Latin America for Life Technologies. You may have seen in your invitation and your agendas that we were expecting Eric Venemeyer to be here. He is the marketing lead for genetic analysis. Unfortunately, he had some flight problems and was not able to make it today. So with the help of Sophia and my Argentinian colleagues, I will present for you an overview of Life Technologies genetic analysis portfolio products, looking at how Life Technologies has led the way in real-time PCR and nucleic acid sequencing research over the various decades and generations. There we go. It's the down button. When you talk about genetic analysis, I think one of the first things that comes to mind is the Human Genome Sequencing Project. This is a seminal project where genetic analysis tools and research was actually done. The goal of the project was to sequence the 3 billion nucleic acids that comprise the human genome. That's about 25,000 genes. And it was targeted to be done in about 13 to 15 years. And I think if we analyze the progress of the Human Genome Project, we can get a good understanding of the evolution of genetic analysis tools and also a really good understanding of how Life Technologies was played an important role in the development of those tools and the evolution of genetic analysis as a technique. And what I'd like to do is start off by sharing my personal experiences with you. I was actually one of the researchers on the Human Genome Project at Washington University in the United States. Now, like some of you in the audience, your introduction to sequencing and genetic analysis was probably through radio-labeled technologies. That's dating myself up here. When I first started to do sequencing, I used radio-labeled technology and I sequenced a 3,000 base pair gene, and it took me about 3 years. So that's an average of 1,000 base pairs in a year. Now that's how you're probably a new sequencer because your eyes are very wide. That sets a baseline for you to understand the evolution of the technology. When I started on the Human Genome Project, we were in the pilot phases. We were proving proof of principle. And we were sequencing C. elegans, which was a nematode. And at that time, Applied Biosystems had just introduced fluorescent labeled sequencing. So they had fluorescent chemistries to incorporate a dye into the nucleic acid sequencing reaction and instruments to detect that fluorescent chemistry. And at the time, we were doing slab-based gels. We did 24 lanes on a 26 centimeter gel, generating about 200 to 300 base pairs per sample. So now we're sequencing in a few days a couple of thousand base pairs. As the technology progressed and the project evolved, we went to 36 centimeter gels, getting longer reads. We went to 36 samples per well, so processing more samples at a time. And as the project neared its completion, we were using capillary electrophoresis instrumentation and generating anywhere from 10,000 to possibly 100,000 samples in a day. Now at the Genome Center, we had about 100 capillary instruments. So that gives you an idea of how many instruments it would take to generate that much data, that much information. And in fact, the introduction of the Applied Biosystems 3700 capillary sequencing instrument was attributed with saving about 5 years of time in the completion of the human genome. Now that was a decade or more ago, and that gives you an idea of how this technology evolved in that period of time. And you'll hear later today how genetic analysis and Life Technologies continue to evolve sequencing applications, genetic analysis applications in the form of two products that we're launching for you here today in Argentina, the Ion Proton instrument and the QuantStudio Real-Time PCR instrument. It's important to note that during this same decade of time when the human genome was being sequenced, Applied Biosystems, which is now a Life Technologies brand, also developed and introduced real-time PCR technologies. They introduced instrumentation to detect fluorescently labeled nucleic acids that were incorporated during a PCR reaction, as well as chemistries that I'm sure you're all very familiar with, such as cybergreen and maybe even the more well-known TaqMan chemistries, TaqMan assays. So this just gives you an example of how Life Technologies as a company, through its involvement with researchers and its collaboration, was critical in the evolution of genetic analysis techniques and supporting that type of research. Now, in addition to being a key player in the evolution of the basic research applications and techniques, Life Technologies is committed to really developing and improving life and accelerating and improving discovery. And in fact, our corporate mission statement includes the terms shaping discovery and improving life. So those are really key to Life Technologies as a corporation and how we want to engage with you, our customers, and the research community, and how we conduct ourselves as a company. And in these next few slides, I'm going to show you some examples of how we as a company and we as an organization engage with our customers and with researchers to do just that, shape discovery and improve life, beyond just building basic scientific techniques. The first example is a breast cancer trial that Life Technologies supported through funding and instrumentation and reagents. It was found that there was a type of breast cancer that really didn't act the way you would expect a cancer to. It didn't express itself the way many breast cancers did. And because of that, it was resistant to traditional screening methodologies and therefore traditional treatments. Through the use of whole genome sequencing, researchers were able to discover the mutations that were causing this change in this particular type of cancer, and they were able to develop appropriate screening techniques which helped identify those women that were suffering from this particular type of cancer and subsequently develop the appropriate treatment for them. So a very clear indication of how genetic analysis tools are impacting people's lives. Excuse me. The next example is a way of how genomic sequencing, genetic analysis is impacting medical treatments and the design and application of particular medicines. In the instance of the Berry twins, at a very young age, these twins exhibited symptoms that were detrimental not only to themselves but to their families in terms of having a normal functioning lifestyle. The twins would just spontaneously pass out, lose motor control, and really it was impossible for them to even leave the home or have a normal life with their family. They went through thousands of tests and millions of dollars in money trying to understand what the cause of this disease was. And they finally approached the Baylor College of Medicine and had the genome of the twins sequenced. And a mutation was identified that was causing these symptoms within the twins. And it turned out that a very simple drug treatment, it's about $40 a month in pills for these twins, eliminated the symptoms, and you can see them now, and they're having a very active, functional, normal life. And that's all as a result of whole genome sequencing and tools that were provided by Life Technologies. The last example I'm sure many of you are familiar with, as recently as last year, there was a fatal E. coli breakout, a mutant strain of E. coli in Germany. And researchers using Life Technologies' PGM system were quickly able to sequence the genome of the E. coli within a matter of days after having sequenced the genome, develop a screening assay that helped people identify the source of the E. coli and control an epidemic in Germany, a fatal epidemic, that could have turned into a pandemic in just a matter of days. So I think that's a very clear example of how genetic analysis tools have evolved and how, again, Life Technologies is helping to support the research and the applications that these tools can be applied to. These next examples, we're looking at areas that aren't necessarily related to health or medicine, but really more to improving the human condition in general. Whole genome sequencing applications are being used to study plants as a potential source of biofuels and improve feedstock. We all know that looking at biofuels, alternative energy sources, is going to have a huge impact globally someday. So this is key research that impacts everyone. The second example, the CE technology is being used much more prevalently on a day-to-day basis for human identification applications. And not only from the aspect of understanding victims of crimes or unknown people, people that are lost, et cetera, but also from the perspective of being able to help identify criminals, or even more importantly, exonerate people that may have been wrongly accused or in some instances wrongly imprisoned. So the availability of DNA samples and the technology now with CE chemistries and CE applications allows this to happen. And there are many stories coming up in the past couple of years about people that have been exonerated after being wrongfully accused and imprisoned. The last example is looking at crops and food sources. Using genetic analysis, real-time PCR, and sequencing technologies, people are able to better study crops and food sources. For example, you can develop drought-resistant plants or plants that offer more nutritional value as a food source, and use these in areas where they have the need and they have nourishment and feeding problems, such as the sub-Saharan areas in Africa. So again, these are just examples of how genetic analysis tools and how Life Technologies' engagement in developing and innovating these tools is having an impact not only on health and medicine, but really the human condition overall. Again, aligned with our mission statement, we are shaping discovery and we are improving life. And it doesn't stop there. Edward, could you grab the water for me? Thanks. It doesn't stop there. I mean, Life Technologies, we've innovated over the past couple of years, and thank you. And we continue to innovate and continue to work with people such as yourselves to understand the needs in science, where you need science to go, where humanity itself needs to see developments and improvements. This is a quote in one of the recent technology reviews about the Ion Proton instrument that you'll hear about today. Thank you. And as a company, again, with our mission statement of shaping discovery and improving life, we as a corporation are committed to continuing to get quotations such as this and to get notice by you, the researchers, as well as those that review and understand technologies. This next slide gives you an idea of the scope of Life Technologies' commitment and approach to understanding science and supporting science. This is just a quick snapshot of collaborators, companies that we work with, that we support through products and technology. You can see that we've got academic institutions, pharma and biotech, government labs, public health organizations. As a company, Life Technologies supports research, innovation, and technology around the globe. And there are even some companies here, some of you may even work at them, that we support in Argentina. So this slide just reinforces, again, our commitment to shaping discovery and improving life. Now, when you look at these researchers and these people that we collaborate with, you can see that they have applications across all types of science. We talked about basic research and how Life Technologies instrumentation and reagents have been used to support genetic analysis, sequencing, et cetera. So the basic research areas are covered. Moving into translational research, biomarker discovery, drug discovery, we have tools, we have applications here. And again, as a company looking to the future, getting involved in molecular diagnostics, regenerative medicine, we are always evolving, we are always collaborating with you, understanding where you need to go and where the tools that you need are going to be best applied. Now, I'd like to revisit the concept and the structure of Life Technologies. Life Technologies is a relatively new entity. Those of you that have been around as long as I have may remember the original Life Technologies. But Life Technologies as a company is fairly new, about 3 years old, consisting of the merger and integration of Applied Biosystems and Invitrogen. But in addition to those key brands, Life Technologies is made up of a number of different brands and former companies that you're probably all very familiar with, but may not have known were actually Life Technologies. We have Gibco, we have Molecular Probes, Ambion. Those are all Life Technology brands. Now today, as we talk about genetic analysis, we're going to focus on the Applied Biosystems brand, the TaqMan brand, and the Ion Torrent brand. Now, I'd also like to point out, regardless of what brand product you purchase from Life Technologies, you can be guaranteed you're going to get a high quality product at a great value. And as a collaborative partner for you, our researchers and customers, we are committed to providing high quality products across our portfolio and brands that fit your budget and your needs. Now I'd like to focus now more specifically on genetic analysis. That's why we're all here. We're here to learn about the newest and latest, greatest products for genetic analysis and the tools to support your research. And as a company, when we talk about genetic analysis, we talk about technologies or instruments for speed and cost-effective research, the supporting reagents and assays to help you study your nucleic acids or your proteins, and also world-class service and support that Life Technologies has behind all of the products and instruments that we provide. So what I've done is I've laid out some key applications associated with genetic analysis, and we'll talk about each one of those and use those as a way to frame the product portfolio that Life Technologies offers to support your specific research. You may see yourself up there in some of these techniques and applications. And we'll show you the whole breadth of instruments, reagents, assays, and service and support that we have. So starting with whole genome and whole exome sequencing, this is something that some of you in the audience may be trying to accomplish as the goal of your research. And when you look at what these customers need, they need to identify new genetic risk factors. They're looking at genome screening. They're looking at processing a large sample base. They're looking at whole genomes, lots of data, lots of information. They're challenged by the time it takes to do that. They're challenged by flexibility and the scalability of their instrumentation or their research processes. And I think everybody in here can understand the need for cost and effectiveness in value in what you're trying to do. Even as suppliers of genetic tools, Life Technologies, we're all consumers in one way or another. So we're just like you. We're all looking for the best value for what we're trying to get. And Life Technologies acknowledges this need and acknowledges the challenges that you have. And we are very pleased today to launch for you the Ion Proton sequencing machine from the Ion Torrent brand. We are very fortunate to have Dr. Andy Felton with us today. He is the Senior Director of Product Management for the Ion Torrent product line, and he will be presenting in great detail this new cutting-edge instrument for genetic analysis and genomic sequencing. For those of you that aren't familiar with the Ion Torrent technology, it is based on semiconductor chips, and it's a simple process at its core, but it's a very powerful and effective genetic analysis tool, and Dr. Felton will be explaining it to you in much more detail. There we go. The next area that we want to look at is the genetic analysis application where you're doing targeted sequencing, or applications where you're looking at maybe using or developing a gene panel. Your need here is to rapidly identify mutations or specific genes that you want to study. You're looking at developing or using a gene panel. You're concerned about cost. Again, cost is always going to be an issue. You need accuracy. Since you're targeting an area, you want to make sure that that's the area you're looking at. So you're concerned about accuracy. And in this instance, you also may be concerned about the availability of your sample. You might have precious samples, limited amounts of material to use. So that's going to be of concern. And we are fortunate again to have Dr. Joe Boland here with us today. He's from the National Cancer Institutes in the United States. And I know from speaking to him earlier that having that limited input material, limited source material, is of concern in their research. And he'll be talking to you about how they've used Life Technologies Ion Torrent products to overcome that particular limitation and be able to generate the types of research and data that they need and that you yourselves may need. So for targeted sequencing and gene panels, we have the Ion PGM system and all of the supporting reagents that go with it, the enrichment kit that helps you get around the sample limitations. There's the Ion AmpliSeq cancer panels that provide you that accuracy and targeting that you need, as well as the Ion Reporter software that helps you process the large amounts of data that you have and get you the answers that you're trying to find. Talking a little bit more about the product portfolio and the panels, we have two types of panels available today, what we call the ready-to-use panels, and then also we have custom panels. For the ready-to-use panels, we have a cancer panel, which is about 45 or 46 cancer genes, and then we have the comprehensive cancer panel, which is greater than 400 cancer genes, ready to use, ready to order today. We have the infectious disease panel representing about 100 heritable diseases that you may be interested in studying. And then if you're not looking at cancer or one of these inherited diseases, we have custom options with the AmpliSeq Custom Designer. Now I do want to point out this is a new product. The AmpliSeq Custom Design is a new product for Life Technologies. I'm currently in the process of working with the team in the United States to make it available here in Latin America. So if you have interest or need for a custom panel, please talk with Sophia or your local rep, and we'll keep you up to date on when that particular product will be available for Argentina. It shouldn't be long, a couple of months. Now, when you look at targeted sequencing and using gene panels, this is a really good application area to present to you how the different parts of Life Technologies come together to offer you a comprehensive solution, and how bringing applied biosystems and Invitrogen together and Ion Torrent really provides a best solution and a comprehensive solution for your research. If you're looking at doing a large amount of screening, trying to understand and focus in on a target from a large sample source, you've got your PGM system to do that. Once you've identified your target, you need to do more detailed, more focused research. You move into the real-time PCR side, and you'll learn more about our newest real-time PCR instrument, the QuantStudio 12K Flex, a little bit later today. We have Edward Enright with us from the United States. He's one of our top field application specialists in the area of real-time PCR, and he'll be talking to you in much greater detail about this latest and newest addition to our real-time PCR family of products. The next application area we want to look at would be genotyping. This is where you're looking at a specific gene and looking at variants within that particular gene. You're going to want to, again, be very specific. You want to look at an actual target. You want to detect the presence of the cell-based changes at a genomic level. You're going to be concerned about your input DNA, the amount of sample that you have. And you're also looking at that accuracy and the affordability to detect low-frequency mutants. And similar to the targeted genome approach, the targeted sequencing approach, this is an area where Life Technologies offers a full breadth of products and solutions for you to help support and direct your research. You've got your Ion Torrent PGM to do your broad screening. You can move into your target more focused with real-time PCR. And then you can also look more at a mid-range using the capillary electrophoresis system. So you do your initial screening at a broad range with a PGM. You go a little more focused on capillary and then really fine-tune your target using real-time PCR. Now, I told you how our products were divided into those technologies, the systems, and then the supporting reagents. And it's when you get into genotyping that you see the true benefits of the breadth of Life Technologies products. You're all familiar, I hope, with TaqMan. So when you're trying to do genotyping, you have access to the TaqMan SNP genotyping assays. All of our TaqMan products are backed by a guarantee around quality and performance. We have millions of assays pre-designed, ready for you to order, ready for you to use today. And just like with the AmpliSeq custom products, we have custom designing software so that you can actually find the primers and the TaqMan assay that you need for your specific target if it's not one of our ready available, ready-made assays. If you're not studying SNPs, but you're interested in some other type of single change or some other type of mutation that you want to study, we have pre-designed mutation assays and, again, the ability to do custom designing. All of which the assays can be used on any of our real-time PCR instrumentation, from the introductory step 1 system, which is a 48-well system, all the way through the new QuantStudio 12K Plex that Edward will be talking about a little bit later. Now, I do just want to give you a quick introduction to this system because it is so exciting and so new for us, and it's a great tool for you in your research. You can see here that any type of application, mutation detection, copy number, gene expression, epigenetic type studies, looking at genomic biomarkers, all can be done on this single system. You also see that you can use 96 well plates, 384, or our open array plates, which are over 3,000 assays in a single plate, 3,000 samples. And that's how this system gets its name. When you look at QuantStudio, it's quantitative, and Studio, it's a single instrument that does all of these different applications. It's a suite of applications or a studio of applications in a single instrument. 12K Flex is because it's flexible in terms of the number of samples it can do. It can go from 96 up to over 12,000 samples in a single run using the open array technology. And again, Edward is going to be introducing this great new product to you a little bit later today. Whole transcriptome sequencing and gene expression analysis is probably one of the more prevalent research application areas where genetic analysis tools play a major role. And again, this represents an area where Life Technologies is uniquely positioned to offer you the broadest range of solutions and tools for this particular type of research. When you're doing gene expression or whole transcriptome, you're looking at new RNA transcripts, or you may be wanting to study the expression of known RNA transcripts. Some of your challenges are going to be in the diversity of RNA that you're trying to use, as well as the inherent instability of RNA. As I said, Life Technologies is uniquely positioned here because we are comprised of not only the Ion Torrent brand and the Applied Biosystems brand, but we also have those Ambion products. And those of you that know Ambion know it's the RNA company. So again, Life Technologies is an excellent partner for you to be working with when you're doing these types of studies and looking at RNA. When you study RNA, again, you have a couple of different ways and different things that you're trying to look at. You may want to study known transcripts. You may want to look at novel transcripts, something new and unknown. And again, your transcript source may vary. Non-coding RNAs, microRNAs, small RNAs, messenger RNAs, tRNAs, and any RNA you may want to study. There are a couple of approaches that you may want to take when you're doing RNA expression. You can have your hypothesis-based studies where you have a fairly good idea of how you expect your RNA to express itself. It's a low expressor. It's a complex transcript. These are the instances you have a few genes, you already have a gene panel designed. These are the instances where Life Technologies real-time PCR solutions are going to be the best approach for you. You've got your custom assays that you can design. You've got your pre-designed assays that you can use. You have an idea of how your transcript's going to act, and you put it on one of our many real-time systems. Again, using the TaqMan brand as well as the Ambion RNA products. Now, on the other side of things, you may be looking at a novel transcript. You may have no idea how you expect this particular RNA transcript to act or perform. This is where the Ion Torrent systems really come into play. Due to their scalability, their ease of use, their affordability, you can do large transcriptome screens and large transcriptome sequencing in a reasonable amount of time and affordable cost, and discover and understand that new transcript that you're trying to study. Again, supported by not only the Ion PGM products, but the Applied Biosystems and the Ambion products. Life Technologies has that full solution for you. I talked about the scalability, the simplicity, and the speed of the Ion Torrent system. This just reinforces that. You've got the tools to do your library construction, to prepare your RNA. You've got the scalability of the different chips, which Andy will present later, so that you can understand the processivity and the amount of samples you can run on this single system. And lastly, the speed. You can see here, with 6 hours of prep time, you can have an answer in about an hour for small RNAs or 2.5 hours for your messenger RNAs. So it's fast, it's simple, and easy to use. The last application that I want to touch on for you is epigenetics. Now, with epigenetics, you're not looking necessarily at what's happening with the genetic material, the nucleic acids. You're looking at other effects that make a change to the phenotype based on interactions with the genetic material, the nucleic acids. And even though it's not classic nucleic acid studying, Life Technologies, again, has the tools to help you understand epigenetic changes and how the phenotype is being expressed due to the interactions of the genetic material and other molecules. So your PGM sequencer is useful here, your QuantStudio, and again, going back to the marquee brands or all the different brands that make up Life Technologies, you have your Ambion products, your TaqMan or Applied Biosystems products, and your AB products, all of which can be used to support any type of epigenetic research that you're looking to do. Now, as I said earlier, we have our technologies. I've shown you the instruments, and we have our reagents. And you see here the full breadth of our portfolio and all the different products and systems that we have to support any type of genetic analysis research you want to do. On the discovery side, you have the Ion Torrent products, large-scale screening, large-scale sequencing applications. As you get more specific, looking at characterization, you move into the capillary electrophoresis or the real-time PCR instruments. And again, all of these are supported for your screening with the wide range of products, the TaqMan assays, either pre-designed, customizable, as well as the AmpliSeq pre-designed and customizable panels that you can use. All of these products and systems are backed by Life Technologies' world-class service and support. And this service and support takes many forms. When you purchase an instrument from us, you can get on-site training. You could take a trip to one of our training centers. You can get online training. We have field application specialists that can come to your lab, help you set up your instrument, help you optimize and design your protocol. We have local technical service here in the Argentina office, as well as access to even more technical support through our experts in the United States and even around the globe. And as today you have the opportunity to talk to some of our leading internal people like Edward and Andy, as well as the opportunity to network with colleagues such as Joe to share your experiences, your ideas, and find out how they leverage the information that they have and their experience with genetic analysis tools and information. So just in conclusion, comparing genetic analysis tools and applications across the generations. Life Technologies was there when the basic TACMAN real-time PCR reactions were introduced. And we continue to drive innovation and to collaborate with you and understand science to the point where we have a quantitative multi-application system like the QuantStudio 12K Flex that you'll hear about from Edward. We were there to introduce Sanger sequencing and move us out of the radio-labeled age of sequencing into fluorescent dyes and labels and continue to do that with next-generation sequencing. You'll hear about the Ion Proton. As I told you, it can process a genome in a day at a reagent cost of about $1,000 US. And as a company, we're committed to shaping discovery and improving life and being collaborative with you, our customers, and with researchers around the globe to keep driving the future and the innovation of science. As a company, we're looking at molecular medicine, regenerative medicine. We're starting to look at single molecule sequencing, the next generation, if you will, of sequencing. So just remember, Life Technologies is a company, while we're relatively new as Life Technologies, we've been around for a long time. We span the generations. We are there with you as science evolves and develops, and we will continue to be there with you in the future. So I thank you for your time and attention. That gives you a brief overview of where we are with Life Technologies. As Sophia said, the rest of the day is really going to be spent probably more on what you wanted to hear, the deeper technologies and the introduction of these two new great products, the Ion Proton and the QuantStudio. So I appreciate your attention, and I think right now we're going to have a short break, right? So Sophia will move you on. Thank you very much.